Okay, thanks very much, Joanna. We now canter on to Malcolm Macleod, who is very unusual in the sense that, well, several ways, but in the sense that this occasion, that he's an NHS consultant neurologist for a lot of his time in Stirling, and he's also a senior lecturer, and he has done both clinical research and he has also done animal research in a lab. So if anyone can understand the interconnection between these things, it should be you, Malcolm.
Well, thank you, Charles, for those kind words of introduction, and thank you for the invitation to speak. What I want to do today, as Charles said, is canter through some of the difficulties that we've had in translating efficacy from animal models of stroke to the patients that I see in my clinic with stroke. The neuroprotective dogma is really pretty clear, and it's probably known to all of you. The dogma is that when you make a neuron ischemia, lots of bad things happen. Some of these things are active, some require energy, a lot of them require enzymes, and so these are things that potentially at least you could block happening. If you blocked them happening, you could save the nerve cell. If you saved the nerve cell, you could save some brain tissue. And if you saved brain tissue, you could improve the functional outcome for those 20,000 patients a year in Scotland who have a stroke, those 5 million patients a year around the world who die of stroke, 400 a year coming to see us in Forth Valley. So a big problem, and one worthy of our attention. And indeed it has received a substantial amount of attention. These are looking at experiments reporting the results of various neuroprotective interventions in cell culture or animal models of stroke from the very first experiments in 1955, round about the mid to late 80s when I was finishing my pharmacology BSc. There was one publication a day on neuroprotection, and that has increased. So now there's about 3 a day. And so where has all of that got us in something that I can give to real patients in the real world to stop them becoming dead or disabled after their stroke? Well, with colleagues in Melbourne, we did a systematic review of neuroprotective interventions, and we found descriptions in the literature describing outcome for 1,026 interventions in different models of stroke, be it cell culture or tissue slice or focal ischemia models. 603 interventions tested in focal cerebral ischemia, largely middle cerebral artery occlusion modelling in the rat, but other species as well, of which 374 of those interventions improved outcome, either as infarct volume or as a neurobehavioural score, in those animal models of ischemia. Not a lot of them have been tested in trial. When we did this, 114 had been tested in clinical trial. You can add at least one NXY059 to that list. 97 of those had come through animal experiments, but for 17 of them we couldn't find any published information regarding their testing in animals whatsoever. And if we look at the ones that I can use just now in the clinic, there are 3. And this one's not even a neuroprotective drug. This is tissue plasminogen activator, which lyses the clot which causes the stroke. And these 2 are aspirin and care and rehabilitation in the stroke unit. So the effective drugs that we have for stroke haven't come through this translation, and we can add another one to the 94 here, sorry, the 96 here that didn't work, NXY059, supposed to be the next big thing, didn't do anything. So what are the plausible reasons? And I think there are only 3, in general terms, 3 plausible reasons for this failure of translation, and I use the word because it tells you what I'm talking about, for this failure of translation. Firstly, the animal studies may have been falsely positive. That's to say, they may have reported an efficacy in animals where indeed there was no true biological efficacy. Secondly, the clinical studies might have been falsely negative, that they didn't test the drug in the right patient at the right time with the right outcome measures, and given a fair wind in the right circumstances, the drug would have worked in these clinical trials. And thirdly, animal studies might not model human stroke with sufficient fidelity to be at all useful in the development of stroke drugs. Now, each of these would be a serious issue, and we've been doing some work on the first 2 of these, and I'm going to talk a lot about animal experiments, not very much about clinical trials, and I'm not going to mention at all this last bit. So one of my very early exposures to neuroscience community was the Society for Neuroscience conference in Miami, Florida, in 1997, I think, where I saw a poster of this work that went on to get published. And this was great, and I spent hours trying to argue the man out of this. This is testing low-dose glutamate in focal cerebral ischemia. And this is outcome measured as an infarct volume. This is control condition, half-dose low-dose glutamate, full-dose low-dose glutamate. And what he hasn't told you is that full-dose low-dose glutamate is 10 to the minus 60 molar. So either homeopathy exists in the laboratory of the Walter Reed Army Institute of Research in Washington, or there's something wrong with this study. And I would like to show you that I think there's something wrong not just with this study, but with a lot of our other studies. So 4 years ago, this chap in the States looked at the abstracts that were submitted to one of the Emergency Medicine Society congresses in the States, and he divided into the abstracts into those that reported that they'd randomly allocated animals to their group, and those that reported that they blinded the assessment of outcome. And what he showed, really beyond any doubt at all, was that those studies that didn't randomise and didn't blind were much more likely to report positive results than those which did, suggesting there's something about the way the experiments were done which were confounding the results. And then more recently, Bart van der Worp and colleagues from Utrecht looked at experimental stroke studies. There are... That's why you're not allowed a phone near a ventilator in intensive care. It's not quite. If it's a neurology referral, Charles, take it, and you can discuss it with me later. So there's the Stroke Therapy Academic Industry Roundtable, who set out criteria for how drugs should be tested in stroke and what quality that testing should reach. And what Bart showed was that the experimental studies performed atrociously against these published criteria that were the in-house published criteria. So against that, what we did was we sat down to try and work out what quality issues we might expect to see experimental work in stroke have. Some of those were things that we thought were important. Some of these were things that other people thought were important. And some of these were things that we knew were important in clinical trials and might be important in animal studies. And so this is our 10-item checklist. Publication following peer review, because that's said to improve study quality. And I'll show you something a bit later on which calls that, I think, into some question. Controlling temperature of the animal, because we know that hypothermia may be protective. Randomization. Allocation concealment is when the scientist who's inducing the stroke doesn't know whether the animal's in the control group or the treatment group. And blinded assessment of outcome is when the scientist calling infarct volume or calling the animal behaviour doesn't know whether the animal got drug or got control. And then there's a number of other things in here as well, but those are the major ones. Now, we came to this not because we were kind of scientific eco-terrorist types trying to give everyone a hard time, but because we were trying to pick a drug from the animal data to take into a clinical trial in stroke. So we were setting out to organise a clinical trial in stroke in the ambulance and try and say, well, how can we maximise what we gain from the animal data? And we did this using a technique called systematic review and then meta-analysis, where instead of just taking those those publications in Nature and Neuron and Cell, we try and get all of the evidence and put all of the evidence together to see what all of it says together. And so this is a forest plot, like Joanna showed, except it's on its side, because I'm a pharmacologist first, I think, and a meta-analyst second, probably. Where the higher up you are here, the more effective FK506 is. This is the line where it doesn't do anything before here. FK506 makes things worse. And these aren't standard errors, and they're not standard deviations. These are the 95% confidence limits of the estimate. And this grey bar here is what FK506 does altogether, all-comer studies in stroke. But we've grouped these according to the number of quality items on our quality score, which each of these publications meets. And what you see is that high-quality studies give low estimates of efficacy, and low-quality studies give higher estimates of efficacy. And if you were to regress this line, when you would get to the perfect study, FK506 would make things worse. And this just isn't just FK506, because when we look at tissue plasminogen activator, the only drug which we know works in human stroke that's been tested in animals, in the animal evidence, again, low-quality studies give higher estimates of efficacy than high-quality studies. So this is suggesting that there's something about the way these experiments are done and the way they're reported which biases the results and serves to overstate how good these drugs are. Can we start then to unpick what individual aspects of study quality might be driving these differences? This is a systematic review of hypothermia in animal models of stroke. I should say that this involves over 100 publications, over 300 individual experiments, and I think something like 4,000 experimental animals. So it's a sizable body of data from which to draw conclusions. If we split these hypothermia experiments into those which did or did not randomly allocate the animals to the treatment group, you can see that those which did not randomise overstated efficacy by an absolute 10%. So a relative overstatement of neuroprotective efficacy of about 33% simply by not randomly allocating animals to group. In fact, we wrote out to the people to say, you said you randomised, how did you randomise? And got one rather perplexing return saying that animals were chosen at random by the investigator, which isn't quite what we mean by randomisation. So there's probably some dilution of this biological effect. And if we look at the blinded assessment of outcomes similarly, you can see that unblinded studies overstate efficacy. Now, what we've been trying to do is to put this together in a way that we can work out what are the important things that we really need to change and what are the things that really don't matter that much. And so we've been trying to build a regression model. What I'm going to show you is some preliminary data, and we've just got CSO funding to do this properly. But the preliminary data says, let's do a thought experiment. Let's take normal saline, which does nothing, we think, and let's see what the model says an inert biological substance would be reported as doing in a focal ischemia model to infarct volume if you remove the allocation concealment. And the model says it would improve outcome by about 10%. If at the same time you unblinded the assessment of outcome, then it means that the methods that we conventionally use in our focal ischemia experiment introduce a bias of a 20% improvement in outcome. If you use ketamine anesthesia, and ketamine, as you know, is a non-competitive antagonist at the NMDA receptor, then you improve outcome by 40%. And if you use healthy young animals, like what I think I just about still am, rather than the hypertensives or diabetics who reflect the patients that I have who get strokes, then you can reduce infarct volume simply by the bias inherent in your experimental approach by over 50%. And this would be orders of magnitude higher than most drugs which have been taken into clinical trial. So let's go back to our low-dose glutamate with a reported improvement in outcome of 36%, and when you put it through the regression model, which I've just described to you, perhaps unsurprisingly, homeopathic glutamate does nothing. But this take-home serious message here is that I could show you a graph like this for NXY059 or for MK801 or for citicoline or for any number of drugs, which are drugs, proper drugs, like we understand them, which are probably equally susceptible to bias. So I said I'd tell you something about how good the peer review process is. What we did was we looked across 6 systematic reviews, over 250 publications, to see how many of those 10 quality items, and this is percentages, were met. And you can see that most of them were published following peer review, and most of them did manage to say they controlled the temperature. But only about a third randomly allocated to group. Only 1 in 9 had allocation concealment. Less than a third had a blinded assessment of outcome. And only 3% said before they were going to do the experiment how big the experiment was going to be. And of course, there's always a danger. I know this from my old days in the lab, that you do 6 experiments and you get P of 0.07. Just add a couple of animals per group, you'll get there in the end. And of course, you do need to say how big your experiment is going to be before you start, and if it's not significant and you think it should be, you need to start again from scratch with a larger group. And it makes much more sense to work out how big the experiment has to be before you start. Now, of course, this may all be just some peculiar peccadillo of the strange people who do preclinical drug testing in stroke, but I'm afraid it's not. So this is data from Ling Chan, who's a medical student who's been working with us, and what she's done is looked at 200 publications reporting the effect of transgenesis on infarct volume, where you change the genotype of an animal to either overexpress or underexpress a gene of interest, and then look at the effect on infarct volume. And I'm very pleased to say that with Maggie and Karen Horsburgh and others, we've just had our transgenic animal overexpressing MR accepted for publication after tens of thousands of pounds of the Wellcome Trust's money and many person hours of our time and much grief along the way. And I'm very, very glad that it's got a phenotype. I'm not half as glad of that, though, that I am that we actually did conceal the allocation of the genotype of these animals to the person who was inducing the ischemia, and that the outcome assessment was blinded, seen in 8% and 3% of these transgenic stroke studies, respectively. None of them doing a sample size calculation. I mean, I think the amount of our time and energy that's invested in transgenic studies really behoves us to do these studies to the highest possible standards, and it's my guess, based on what we know from drug studies in stroke, that a substantial proportion of transgenic studies in stroke falsely report a protective or exacerbating phenotype. I mean, that's what this data tells us, I think, without very much doubt. So, That's why the animal side of me is terrible and horrible. Can I tell you why the clinical trialist side of me is also horrible and terrible? And that's because clinical trials, I think, may well have been falsely negative. This is very briefly a tale of two drugs, tissue plasminogen activator and Terilizad, a 21-amino steroid. Both appear to work in animals. We've done systematic reviews of both, and they're both effective. TPA works very well in humans, and Terilizad makes outcome worse in humans. If we look at the median time to treatment in the animal studies for Terilizad was 10 minutes. That's to say the animal had the stroke, and the median time to giving the Terilizad was 10 minutes. For TPA, they had the stroke with a median time to treatment of 90 minutes. Move forward to the clinical trials. Terilizad, a median time to treatment of 300 minutes, 5 hours, with a maximum time to treatment of 24 hours for a drug where the vast majority of the animal evidence comes within the first hour. And TPA, median time to treatment of 90 minutes. So if you look like for like at efficacy with time to treatment, these are the animal studies, these are the human studies. This is a meta-analysis of most of the human studies of TPA. This is a meta-analysis of, we think, all of the animal studies of TPA, with TPA delivered in the first 90 minutes, the second 90 minutes, the third 90 minutes. And you can see this fall-off of efficacy is, I think, almost identical for each. We use TPA routinely in the first 3 hours, and trials are ongoing in the second 3 hours. So there's an argument that says that if actually you have concordance between how you test these drugs in humans and how you test them in animals, you're much more likely to at least give the drug a fair chance to work. Whereas with Terilizad, the drug never had a chance to work testing it under those circumstances. So to wind up, I think animal experiments probably have been falsely positive. I think clinical trials probably have been falsely negative, and I think as long as we have these two faults, we can't say anything sensible about the fidelity with which animal studies of stroke model the human condition. There are a large number of people involved in this in a collaboration which now goes around the world. And for those of you who are interested, we're having a BNA symposium here in Edinburgh up at the Royal Society in October. Thank you very much for listening.
Right. Thank you very much, Malcolm. I'm sure most of the audience will be very cheered by that particular message.